The soma, place somata or somas, perikaryon, place perikarya, neurocyton, or cell body is the bulbous, non-process portion of a neuron or other brain cell type, containing the cell nucleus. The word soma comes from the Greek soma meaning body. Although it is often used to refer to neurons, it can also refer to other cell types as well, including astrocytes, oligodendrocytes, and microglia. There are many different specialized types of neurons, and their sizes vary from as small as about 5 micrometers to over 10 millimeters for some of the smallest and largest neurons of invertebrates, respectively. The soma of a neuron, i.e., the main part of the neuron in which the dendrites branch off of, contains many organelles, including granules called Nissl granules, which are composed largely of rough endoplasmic reticulum and free polyribosomes. The cell nucleus is a key feature of the soma. The nucleus is the source of most of the RNA that is produced in neurons. In general, most proteins are produced from mRNAs that do not travel far from the cell nucleus. This creates a challenge for supplying new proteins to axon endings that can be a meter or more away from the soma. Axons contain microtubule-associated motor proteins that transport protein-containing vesicles between the soma and the synapses at the axon terminals. Such transport of molecules towards and away from the soma maintains critical cell functions. The axon hillock is a specialized domain of the neuronal cell body from which the axon originates. A high amount of protein synthesis occurs in this region, as it contains a large number of Nissl granules, which are ribosomes wrapped in RER, and polyribosomes. Within the axon hillock, materials are sorted as either items that will enter the axon, like the components of the cytoskeletal architecture of the axon, mitochondria, etc., or will remain in the soma. In addition, the axon hillock also has a specialized plasma membrane that contains large numbers of voltage-gated ion channels. Since this is most often the site of action potential initiation, the survival of some sensory neurons depends on axon terminals making contact with sources of survival factors that prevent apoptosis. The survival factors are neurotrophic factors, including molecules such as nerve growth factor (NGF). NGF interacts with receptors at axon terminals, and this produces a signal that must be transported up the length of the axon to the nucleus. A current theory of how such survival signals are sent from axon endings to the soma includes the idea that NGF receptors are endocytosed from the surface of axon tips and that such endocytotic vesicles are transported up the axon.
Here's a neural model. As we take a look at these little parts of the neuron, let's get oriented first. Over here on this side would be the body, as well as we come down, this would be the distal portion, or the axon. So let's begin to focus on some of the structures that we can see here on the body. Well, first things first, we can kind of just say that that whole thing is the body, or also known as the soma. Soma means body, or also known as the perikaryon. So the perikaryon, people have defined it as, oh, that's the area around the nucleus, which would be the body. So that's why we call it that. So it could be the perikaryon or the soma. Most people call it the soma. If I were to lift this off, they're just trying to illustrate that, yes, it's three-dimensional, but if I were to cut this off, you can see that it looks very similar to a normal cell. You got the nucleus, you got all these endoplasmic reticulum with the Golgi body kind of here hanging out, and then all these little red dots, those are all the ribosomes, making all our wonderful proteins. So let's kind of get a couple things under our belt before we move on. If we broke out the neuron into 4 major parts, you can divide them into the things that come off the body, which are called dendrites, the body itself, the soma, or perikaryon, the axon that goes all the way down, and then the part that we actually don't see is when the axon then splits off into its axonal terminals or synaptic terminals or boutons, things like that, whatever you want to call it. But that part is not on this model. We have these 3, dendrites, soma, and then the axon. All right, let's take a look at some of the other additional structures that connect this. If a neuron is in space, it is useless. I mean, neurons are made to carry information, and it's just like saying if I have like a video cable or a computer cable and it's just sitting on my desk, it doesn't connect to a computer, it doesn't connect to a TV, it doesn't connect to anything, it's useless. So any kind of neuron has to be connected to something else, whether it's a muscle, whether it's the brain, or something it has to be connected. And here is demonstrated with plenty of other connections. So you can kind of see these other little things that hang off, and touching these dendrites are from other neurons, making connections, passing information. So these are our axonal terminals or synaptic terminals, and then in between would be our synapse. Synapse is just space between the cells. All right, let's get to the nitty-gritty of the anatomy here. So if we already got the dendrites, the soma, the axon, well, what else can we see? We can see the nucleus, the Golgi body, and then this rough endoplasmic reticulum. These are called Nissl bodies. When somebody kind of looked at it, they saw, wow, there's so many kind of ribosomes hanging out from there. Well, that person was Mr. Nissl, and he says, they're my bodies, so I would call them Nissl bodies. I don't know if he had that accent or not, but regardless, that is Nissl bodies, like the stuff that you can kind of see on a microscope. They look like that. They're the ribosomes with the endoplasmic reticulum kind of hanging out from there. So in addition to that, all these dendrites come together in the soma, and then there's only one axon coming out. So this part right here, you kind of see it cut away, leading into this very, very long axon. This would be considered this triangle portion called the axon hillock, or the beginning of the axon. Beginning of the axon is the axon hillock, most sensitive part of the neuron. Specifically, when we say sensitives, it just has the most channels, so that things can be stimulated there, and voila, you can stimulate the action potential all the way down.